Now let's talk about temporal characteristics of vegetation. Timing is very important when you're trying to identify different types of vegetation. Why is timing so important? Because for different types of vegetation, they have different growth cycles. Okay, you need to know different characteristics in different months or different seasons. Okay, for specific species, for specific type of vegetation, to know when is the best time to address that type of vegetation with remote sensing technology. Okay, here we have a figure showing you the life cycle of winter wheat phenology. So you can see that the horizontal axis here is for different months within a year from September to next year's August. And the vertical axis, if there is one, is of course for the height of winter wheat. Okay. So usually from this figure, we know that winter wheat seeds are sowed in September, and for the whole autumn and winter, winter wheat barely grows. Okay, they're very short and they're covered by snow in winter, and they begin to grow very fast from March, which is the very beginning of spring, and during the summer it grows very fast, and in May and June, especially in June, it is ready. When it is in June, it is ready to be harvested. Okay, so according to this figure, we can see that the best timing to address vegetation, to address winter wheat with remotely sensed data is maybe in summer, April or May. Winter wheat should be very green. You say maybe June. June is okay, but you should expect that in June, winter wheat is turning golden instead of green. Okay. So if you're trying to identify healthy, well-grown winter wheat, maybe May or late April is the best timing here. Okay. And for different crops, for different types of vegetation, life cycles, they are totally different. Okay, here we have 1, 2, 3, 4, 5 figures showing you life cycles of 5 types of vegetation. For example, soybeans, corn, winter wheat. Well, winter wheat, we talked about that a little bit, and the cotton, tobacco. They're all crops. They're all closely related to our life, to economy, right? So again, we have different months here, and also the height, the typical height of a plant is addressed by horizontal axis, right? So when you're actually doing a research with remotely sensed data and you care about the specific species of vegetation, you should find a figure or figure out the life cycle of it, just like this, right? To find out not perfect, proper, say that, proper timing to address your topic, okay? And here, so these figures, they're showing you the height of a plant in different months of a year, right? And we also can address the greenness of a plant by month from January to December. So here we have 2 examples. The solid, the thick black curve here is for greenness of cattails. This is cattails. It can be widely found in wetlands. Similarly, we also have another plant here. It's called water lilies. They can also be found in waterlands. Maybe these 2 species, they live together, right? Say there is a very shallow lake, and in the middle of the lake, you may find water lilies, and around the lake, around the lake, there are wetlands and you may find cattails. So in a remote sensing image, how to differentiate? Um, you can use greenness to differentiate them, right? And water lilies is not there from December to March, which means that in winter there is no leaf for water lily. That's the reason why the curve for water lily is not even there for December, January, February, and March. But even in winter, there is cattails, and it's not that green, but it's still there. You can find cattail leaves in winter, right? So here is the difference. Here is the difference. But in summer, from May to October, October is not summer. From May to September, you can find both types of vegetation in a lake and its surrounding area. But the greenness of cattail is higher than water lilies. Okay. And next, I want to introduce several widely used vegetation indices. Okay, you may have seen some of them in this class. You may have seen some of them outside of this class somewhere else, but I just want to give you a brief introduction of them. Okay, so vegetation indices, they are defined as dimensionless, which means there is no unit. What is a unit? Temperature is there are Celsius degree Fahrenheit, right? For distance, there are kilometers, miles, meters. They are units, right? Vegetation indices do not have units, so it's easy to use. You don't have to consider unit issue. And they are radiometric measures that function as indicators of relative abundance and activity of green vegetation. Okay, so widely used one, LAI, leaf area index, total leaf areas per ground area, or simple ratio. I introduced this before, okay, in this class. NDVI is the most commonly used one, even among these commonly used indices, okay, NDVI. And we have, okay, sorry, repeated that. So simple ratio, right? And the moisture vegetation index, MSI, it is used to address water stress of vegetation, okay? And the infrared index is very similar to MSI, and you can consider II as standardized MSI, okay? It's just like NDVI, you can consider this as standardized simple ratio, okay? I will just go through them very quickly, okay? So the first one, simple ratio. The near infrared to red simple ratio is the first true vegetation index. Okay, it's just simply the ratio between near infrared and red, because the vegetation has very high reflectance at near infrared band and the low reflectance at red band. So the higher the abundance, the higher SR value. And NDVI here, okay, the generic normalized difference vegetation index, okay, still it is using near infrared and red band to address abundance of vegetation, just like SR. But it is called normalized because the denominator of NDVI is the sum of near infrared and red. It is used to standardize this process of quantifying the abundance of vegetation. Okay, of course, again for NDVI, the larger the value, the higher the abundance of vegetation, and the NDVI varies from -1 to +1. And usually, if it is larger than 0.3, we consider that this pixel actually has vegetation inside it or within. But above 0.3, the higher the more abundant the vegetation within that pixel. But again, 0.3 is just a widely adapted threshold. Okay, it's not a rule, a law, or something. And the MSI, it is actually a type of infrared index. Okay, because the numerator and denominator of MSI are both about the reflectance within infrared. The difference is that the denominator is near infrared reflectance and a numerator is mid infrared reflectance. Okay, if you get back to the very beginning of this lecture, we talked about the role of water in vegetation in terms of reflectance, right? Water has very strong absorption in mid-infrared band. So if a vegetation, if a plant is very healthy, it doesn't need any more water. The water is very good. There is no water stress. Then MIR reflectance should be high or low. Should be low, right? Because there is abundant water and the water absorbs a lot of energy in mid-infrared. So on the other hand, for near-infrared, water does not absorb that much energy within near-infrared. So if the plant is very healthy and the water is good enough, is abundant enough to this plant, which means there is no water stress, then MSI value should be low because the MIR reflectance should be low. Okay, that's a very general, very simple analysis for this specific MVI, moisture vegetation. So it is simply using MIR reflectance and near infrared reflectance to address water stress of a specific plant or a specific type of vegetation. Okay, so if we standardize MSI, we have another index is directly called infrared index, II. So again, similarly to MSI, it is also using near infrared and mid infrared to address water stress. So what is water stress? If there is not enough water for vegetation, for a plant to stay healthy, then we say the water stress for this plant is very high. If water is good enough, there is enough water for this plant to stay healthy, then we say there is no water stress. Okay, so II is here to do the similar thing. But we say that, we talked about that, if water stress is low, then MSI should be low because the numerator is low. Abundant water, low MIR reflectance. But here, if a plant is very good in terms of water, there is no water stress, then MIR should be low, right? Then the numerator should be large because it's the difference between NIR and MIR. And if MIR is very small, is very low, there is enough water, then the numerator should be large, then II should be large, right? So for II, if a specific species of plant, a type of plant, does not have very strong water stress, then II should be large instead of small. So it is the opposite of MSI in terms of value, the meaning of value, okay? The larger the MSI, the lower the water stress. Oh, sorry. The smaller the MSI, the lower the water stress, right? But the higher the II, the lower the water stress. Okay, so there. different from each other. And for the last part of this lecture, I'm going to give you, say, a very famous, say a project, or you can say this is a community for the study of vegetation with remotely sensed data. It can also be considered as a data source. You can download data from here. Okay, it's called GAP. Gap Analysis and Biodiversity. GAP stands for Geographical Approaches to Planning. So it is supported by Biological Resources Division of United States Geological Survey. And here is the website. So what is it? So the objective of this project is to keep common species common. Okay, so methods identify those species and plant communities that are not adequately represented in existing conservation lands, which means that they want to find out more areas need to be preserved. Okay, and give land managers, planners, scientists, and policymakers the information they need to make better informed decisions when identifying priority areas for conservation. And this is especially important today in the context of fast urbanization around the world, right? When you plan to expand the city, you need to know where you should not go, where the city should not go. Even the city is going that direction, and there is vegetation community should be preserved in that direction. You should just bypass it. Okay, and advantage, GAP analysis is superior to a species by species approach because it identifies and protects regions rich in habitat. Therefore, the animal species that inhabit them can be adequately protected. So here you can see that the focus of gap analysis and biodiversity is actually animals instead of vegetation. But since vegetation plays very important, crucial role in animals' life, so we need to preserve the vegetation, preserve specific regions to make sure that the biodiversity can sustain. Okay, so here the special part about gap analysis is that it does not focus on a specific species or some species. It focuses on the areas, the regions in which those species live, right? So a typical gap analysis includes the creation and analysis of 4 primary GIS layers. That's the reason why I said that you can consider also consider GAP analysis and biodiversity as a data source, because you can download data from there. So several layers here. Firstly, the distribution of actual vegetation cover types delineated from satellite and aircraft remotely sensed data and ancillary data. So this first layer is very close to to vegetation remote sensing, because you can say the data is from satellite sensors and aircraft sensors remotely sensed data, and the necessary ancillary data, for example, meteorology data, climate data, right? And maybe for specific purposes, necessary ecological data, okay, for the distribution of vegetation cover types, which means classification of different types of vegetation. And land ownership, this is administrative data, right? Land management status, the current management status of land. These 2 layers, they have nothing to do with vegetation research. But like I said, sometimes when you are talking about vegetation research, it's about ecology. It is about the conflict between human beings and vegetation and natural lands. So the first layer is about vegetation distribution. The second, the third layers there about the conditions, or we can say the spatial distribution of human beings. Okay, and land ownership, not necessarily only for urbanized areas. Some nature lands, they're also owned by different people, by different stakeholders, but other two layers, they're here. And also distribution of terrestrial vertebrates as predicted from the distribution of vegetation and in situ observation. Okay, so you can go, if you're interested in it, you can go there and play with it a little bit. Okay, it's here just as an example for scientific project or a scientific community, okay, for vegetation remote sensing. Okay, so this is lecture 10. Okay, it's for vegetation remote sensing. We still have one lecture to go, and it will be about water remote sensing. And I will see you in the next and the last lecture of this class. Okay, thank you.